Okay, great. Good morning. This is a week 12 lecture about manifold and tensor. So this relatively advanced topics. Well, advanced is relative to vector or matrix or non-linear objects. But here, manifold is a mathematical object dealing with this nonlinear shape. And the tensor is a high dimensional matrix, basically. So there are many applications in single cell or in general machine learning algorithm. So we are going to cover these topics today. Okay. Okay, so first we will talk about how can we use manifold learning and using embedding algorithm to do the dimensionality reduction. And then once you have two objects, and we are going to do manifold alignment, okay, just put them together, just like a CCA, and we try to find common axis to allow them to share the feature on the same space after embedding into the same manifold reduced dimension space. Then we will look at some applications of manifold learning. Then the next one will be tensor and the related concept is especially tensor decomposition. And then some applications on tensor decomposition. Okay. So what is manifold? This is a mathematical concept that describes a topological space, which is locally Euclidean, but not on the global space structure level. It's very useful if you have this concept on your mind and then you define your algorithm, follow this topological space and respect to local Euclidean properties, but keep in mind that after anything beyond local will not be Euclidean. That's the concept. So this global structure could be complicated. But if you look at the point of this structure, look at its neighbors, and it follow the Euclidean distance definition. So you can describe it using the regular distance measurement. Yeah, so manifold learning can be thought as a generalized linear framework, which is generalized from a PCA and MBDS. Well, those methods are sensitive to local to non-linearity in your data, but manifold learning try to generalize this requirement and apply to nonlinear structure of your data. Okay, just like PCA MBDS, those algorithms are unsupervised. So here is what I mean by local Euclid. Okay, so here on your left, this is a high-dimensional space, which you have an object, have a complex shape, global shape. And so you cannot approximate the distance, for example, this point to that point by its Euclid distance, but you have to look at the whole structure of this shape. Well, locally, if you only look at a patch of this small region of this structure, you can use Euclid distance to approximate the real distance between data points, but not across the structure from one place to another once you have this structure more complex. So Manifold learning is trying to learn this shape, okay? Try to reduce the dimension from D, which is bigger, higher dimension, to a lower one. Okay, you project your data point from a higher dimension to lower. So in this case, this D is 3. Okay, this shape where this D is a 2. So you project this to 2 dimension. Well, the relationship between distance between data points are still kept in this spread out of 2D space. Well, you can look at the color of those points. At the 2 ends, those are the blue dots, okay? And they're close to the different yellow, green, and green, which are far away from red. But if you only consider Euclid distance without looking at this structure, you will have a wrong embedding. And you're embedding in this 2D, okay? My screen is 2D, and the distance from the blue point to red, dark red point is very close. Okay, your projection was wrong. But this is a correct through this manifold learning process. Okay. There are many machine learning algorithms taking manifold structure into account. So those are the lists of all the machine learning methods which consider this nonlinearity and using manifold learning. So we will take a look only 2 of them today. One is called isobmap, another is called Laplacian eigenmaps. So the basic methodology is we have to have this concept of mind and then consider this manifold and try to find the local neighborhood structure. So that's the approach. Both, if not all the manifold learning algorithm we will follow, look at local neighborhood structure. In single cells, which cell which are similar to each other if you use Euclid distance. So you can compute local relationship, but for the longer range of distance, then we have to recalculate. Instead of directly using the Euclid distance, we have to follow the local steps. So you approximate the distance by looking at the local steps and add them up. our goal here is to reduce the dimension for single-cell similarity, project cells into lower dimension and still keep their relationship to reflect their relationship in the higher space, higher dimensional space. Okay. Okay, isoMap is published year 2000 in Science. So here is a concept I just described. You cannot compute the distance from this point to here using this dash line. You have to consider the overall distribution, the structure of all other data points, and they create a shape of the Swiss roll. So in order to compute the distance, you have to consider the Swiss roll structure where you can create this called KNN here data network. Then you work from one data point to another, follow this network structure. Then after reduce the dimension from 3D to 2D, and you keep this network structure at your original distance between these 2 points through the 3 small steps of this network, and you basically step away and approach your distance by small steps. That's the solution. So so what is isoMap? Very easy. Okay, isoMap is multi-dimensional scaling. Okay, the difference is instead of using regular distance, it uses geodesic distance to replace Euclid distance. Okay, to compute the distance between data points. Okay, how can you get this geodesic distance? It's through a weighted graph. Graph is this network. So isoMap is classical scaling for MDS based on the pairwise distance between data points. Okay. Difference is it uses geodesic distance, which derived or induced from the neighborhood graph. So the distance between data points used for isoMap is between 2 points at the sum of edge weights along the shortest distance path between 2 data points. So again, we are going to deal with graph theory and Dijkstra algorithm is classical algorithm. If you have a network, you found 2 nodes on the network, you can find the shortest path using that. So in order to using isoMap, we need to first create network, which is a graph for your data points, and then compute geodesic distance from all pairwise distance between data points, and then use this to replace Euclid distance, and then you take regular MDS, get the results to reduce the dimension. If you don't remember what FDS is, remember we have the map between different major cities, and you have distance between cities, and you want to reconstruct the geological relationship between the cities, and put them in the 2D space, and also allow them the distance between them follow the distance matrix is being given. So it's FDS is from a distance matrix to projected position of the nodes. So here is an example about geological distance geodesic with against the Euclid. So if this Euclid distance you consider one point to another, the distance like this, but in reality you have to follow the landscape, and you go uphill and then down there through some valley. So your geodesic distance is much longer than Euclid distance. And on this graph, if you from this point, say A to B, without considering all the structure of the data points other in the neighborhood, you can have a Euclid distance in this blue line. But if you go follow the shortest path, then you have to move from one point to another node, and then at the end you have the right curve as a distance. Okay, in this 2D object, and then you project them onto 2D, 1D, and then this relationship between data points for Euclid distance, it's close by to each other, but from A to C, for example. But if you unfold it, it's supposed to be A to C to cross a long distance. So that's our by default learning process. Have to try to keep your structure in this 2D and project them onto 1D, but still keep the relationship between data points when they are in the higher dimension. Okay, I think that concept is easy. We just know how to apply them, right? Any other questions so far? Okay. Okay. So this is one of the applications. In this case, we compare some of the. I think this from just from this channel. So this is probably some kind of data collected from MRI or EEG, I don't know, but those are the sampling point on the brain, maybe. Okay, so in this case, we have this high dimensional data. Okay, so then you compare A and B, and then with X. See, X is close to A or close to B. Without considering other sample or other patient or control samples, we may look at, okay, this may similar to one of them. I don't know, X close to B or A. But if you put this in the high dimensional manifold structure, and then you realize X may be more similar to B than A. Okay, so just counterintuitive. If you only use Pearson's dissimilarity as a distance, then this distance to B is smaller than A. Okay, so that's more similar to each other. But if you consider that geodesic distance, that this one, these two are more similar to each other. Okay. Okay. Again, summary. This you get geodesic distance matrix that projected up to reflect the higher dimension structure relationship. So you have seen this many times, I believe. Everybody knows what it is, but for isobmap, we need to have a KN first. Then in the KN, we look at the shortest path from one point to another using either this algorithm or this, then do the FDS. So you see this method in the Manifold learning package is always on the top because it's one of the simplest one. So that's in Julia package. So you load this Swiss row example into X, and you fit, and you transform, then you get the projection of Y. So you fit this model using this method, and then you get projected position of those. So KN is graph-based. Another is related, but it's called data fusion. And they use some kind of still network theory. You use some kernel function and define Gaussian kernel apply to weighted graph and translate this weighted graph distance into some kind of affinity. So you know from what point diffuse the probability to get into another point, what's the likelihood. So you replace the distance using some kind of probability. But essentially, to me, they're the same. They always neighborhood-based method. That's a solution for manifold learning. Okay. Next method we are going to cover is called Laplacian eigenmap. I believe you already know Laplacian, and I had the Blackboard demonstration that's L Laplacian matrix is the degree matrix divided minus W or A adjacent matrix. So the same thing applied here. So this published in 2003 the paper to do dimensionality reduction for data representation, but it's applied for high dimensional and also using manifold embedding to deal with. I'm trying to find the keywords nonlinear. And you see the geometric they motivated and computational efficient, locality preservation, nonlinear. So all the keywords I try to identify as present in this abstract. So this is a method which you use neighbor graph, and then apply Laplacian this graph learning theory and to minimize to minimize cost function based on the graph to ensure the neighbors point close to each other on the manifold are present in the low dimension space to preserve their local distance relationship. It will use eigen decomposition for Laplacian matrix. So that's a summary of this algorithm. We will construct a weighted graph, and we will then select a distance function. You can either you have, if they're connected, you put 1, or you can use some kind of a transformation, hit kernel to transform this weight. And then here you see you compute Laplacian matrix, this graph, and D is weight matrix, which is the degree of the node. Then the eigen decomposition problem is here. It's so-called generalized eigen decomposition, because we are dealing with 2 matrices, L and D, and that's eigenvectors in the F, eigenvalues in the lambda. So once you have that, you use those eigenvectors as new features for your nodes, so you can find the location defined by those eigenvector. Depends on how many eigenvectors you want to use. You want to use 2, then you project the data into 2 dimension. Okay. Okay, here is a visual illustration of this process. We have data points, in this case 3D and yellow points. Then you want to find a graph. This graph is to link by its neighbors. So depends on how many, what k you want. So k equals 3 in this case, maybe. So every node has 3 neighbors, and make a link. And they will for data 1. So this point is not in the nearest k. So this is the 4th nearest. So the only 3 are linked. Anyway, so you can do this for each data point. Look at the 3 neighbors and make a link. And you create a structure of the network. The link between nodes recorded in this W. So if it's linked, put 1. And that degree is the number of how many partners each. So there are 3 for each. And sometimes because they're shared, you may have yeah. But anyway, so this is k equals 3. The difference between them is degree matrix minus this W, and then you have a Laplacian matrix with a lot of negative 1 around here. Okay, when we have KN, normally we consider symmetric KN. So if you have A connect with B, or B connected with A, so you put a link between them. Well, if you have to use mute KN, then you have to have either A and B are nearest neighbor to each other, then you can make a link. Otherwise, if it's only one direction, then you will not keep that because you require mutual relationship between all the data points by each other to each other. They are neighbors, or you can keep the distance directed near the neighbor. So there are several ways to do but this is the most commonly used. Okay, so once you have a KNN network, you have this weight matrix. Depends on whether you want to use the distance or you just want to have an indicator they are linked. So if you decide to have a distance, so this is a weighted matrix. The next you may try to do this Gaussian kernel to transform this. For example, if you have a one data point I, and then j, another data point, the distance between them, you can use this formula to transform it into the weight. What that means is if you have a x in the middle of here, okay, this shape, then your y, your j, another data point, if it's here, then you look at the z value, and that because it's too far from each other, then this weight become smaller. Or if you have your first point is in the middle, I, and your j is very close, right? And your z value project on this curve is high. So if you have two data points close by, and then you give a stronger weight between them. Whether after they move away from each other, then you weight it down and follow this normal distribution. Okay. So once you have this constructed, if it's a weighted matrix, you look at the distance between these two data points, I to j, is far away, and then the weight after the kernel transformation, this will be a small value. Okay, small value here. Well, the close ones, then you have a stronger value. Okay. Okay, for the degree, you sum all the linked for every node. You look at the linked edges and sum the degree, sum this weight. If it's unweighted, then it's just how many neighbors you're linked with. So that's a degree. So you have D matrix and weight matrix, and the Laplacian matrix is E minus W. Okay. So once we have L and the D, so this matrices will be used to found eigenvector. Okay, to put this system. So what is this generalized eigen decomposition? So this generalized eigen decomposition, the function take A and B, two matrices. Okay, remember this D is different from this D here. So take two vectors in here and give you eigenvector and eigenvalue. So in irregular form, so you have V D eigen A. Okay, so irregular form, you just take only one matrix. So that means is you have A, that you use your eigenvector okay. Basically, it's A as a rotation matrix to rotate V with vector, and this is D, which is a Cartesian lambda times V. So that's the regular decomposition. Well, in this case, we have both A and B. In that case, we have V D again both A and B, two matrices. We want to have A times B D times B times B. Okay. So this is a general form, just put one B matrix in the middle. So here we have to follow this A, this is A, and this is a B. Okay, your two input and give you V become F, and D become lambda. And then in the F, the first one, yeah, for the first eigenvalue, the smallest, if you rank them, will be 0, and then you have 2, 3, and we will use eigenvectors associated with non-zero eigenvalues as a representation of this projection. Okay, so you go from data points, look at the network between the data points distance, make a network, and then you transform until you get this representation. You change the original data in the high dimension to this. You can use eigenvector for second and third, second and third or later. So non-zero eigenvalue related associated eigenvector to represent your original data points. So here you can see, like, I want to use this two. So you have an eigenvector F2 and F3. So use the two dimension to represent original data points. The original can be very high dimension. As long as you can compute the distance between the data points, then you can go to step 1, step 2. This is a small example. We have 4 data points on this 2D space. Then we compute KN for them. So K equals 1 here. And the KN looks like this. So your W matrix unweighted. Is this your degree matrix? Is that so? Node 1 have 3 partners, have 3. Node 2, 3, 4 only have 1 linker. So the weight matrix, the degree matrix D looks like this. The difference of that, and then you take a generalized eigenvalue problem. Okay, you get the first eigenvalue 0, and then the second and third lambda 1 and 2 is 1. So you use a combination of these two to look at its eigenvector, and then you get the position. So the first element position should be 0. So here, 0. Second position, note 2, divided into -3 here. Okay, this is 2, 3, and 4 here. So I tried this example. Everything above is okay, but as it down MATLAB and this Python seems to have the slightly different eigenvalue solutions. What I get is the first position is 0. The second, which note number 2, is minus 0.7 somewhere here. And the third position, 0.2. First position, 0.5. Yeah, but numerically, it's not identical, but the general position is fine. Okay, comparison between PCA and the Laplacian eigenmap. PCA is linear dividing, so you just rotate everything and without changing, without unfold, fold, or disturb, transform any the position for the data points. You just rotate them in the correct dimension and project directly. Well, this Laplacian eigenmap actually changed. You bended the relationship somehow, but the projected and the relationship in the low dimension should reflect their relationship in the high dimension. Then of course, how can you compute is different. This is a generalized eigenvalue problem. Use the smallest eigenvectors to project the data points. Well, in PCA, we found the largest. Yeah, so that's one way to do it. Or you use the covariance matrix the solutions. Okay. So if you apply PCA to this example, this switch row dataset is commonly used in dimensionality, nonlinear dimensionality study. And if you can have a method which can project this correctly, because you have a color label between the distance between data points. So your outcome, for example, this Manifold charting should have separation of these different colors, this rainbow colors. So you see, this is okay, this is okay, this is okay, this is okay. So those are the manifold learning algorithms. But the PCA will not. PCA only linear transformation at the project. The sum of red and blue, and the sum of this yellow, green are to the same location, right? You lost the real relationship between data points when they was in the original high dimensional space. And there are other more complex shape. So you have a switch row and also have these holes in there. Look at different algorithm will project those structure correctly or not. So this is one algorithm. is another. I don't know what their name. Yeah. So this looks really complicated. Okay, applications. So before moving on, any quick questions? So I introduced the general concept and the general solution, which based on the neighborhood of network. And then two examples. Isomap is replace Euclidean distance with a geodesic distance. And then this Laplacian eigenmap is again start from a construction of okay, so this is one of the applications in the package of fate. Okay, so the idea is here, the cells are not independently unrelated. The cells are related by their underlying differentiation relationship. So if you have this original structure between cell populations, and there's some kind of trajectories in there, and then dissociation procedures, single cell RSIC will make this information lost. And then you have this matrix that you try to reconstruct their relationship using network and reconstruct cells relationship, this trajectory analysis. And here you can have a dimensionality reduction applications. And we start from the high dimension embedding and look at their relationship, make a network and transform with the kernel and the cape and trim off those distance related linkers. And then learn from this graph the structure of the original. So if you have this by default learning ideas about their pseudo-type applications, you can help the class tree as you know the relationship between cells. So you can do the denoise and visualization, of course. So Monaco 2 slides, those ones all have some these ideas in there. In the middle of those algorithms, for example, in Monaco 2, and then their geodesic distance get involved here. So the DDR tree is graph-based learning. This especially using the KNN. Okay, so look at the manifold, diffusion, geodesic. So they all have this manifold learning concept behind. Okay, so this is the application of manifold learning in a package called Magic. So the Magic is to do the imputation. So we, in this case, try to learn the relationship between cells, and then after do this diffusion and the process imputing will help you to fill in all the empty spots in this matrix. Empty spots are the dropout. They have zeros, and this assume this cell has those genes should be expressed at a certain low level, but it cannot be detected. But here we use cell-cell relationship to create a network and try to approximate their relationship and use neighbor cells to smooth out those relationship of their gene expression. So you fill in this empty space in the original sparse matrix. And this paper suggests if you use magic, so before you always see, for example, this gene A and B, there's no correlation because cells, a lot of them are either active regarding to gene A or B. So a lot of cells will be projected out to this x-axis or y-axis. There's no correlation between gene A and B. So after imputation, you see this clear positive relationship between A and B. Okay, even though cells do not express A and B at the same time, or their expression was not measured because of the dropout problem. But after you do the imputation, then you recover the relationship between these two genes. So once you have that, you can have other, so those are the data points between each point here is a cell. So you recover, you fill in the empty and you improve this dimension reduction problem and make this embedding of the cells more realistic, and then you can detect the spatial position of the cells, see whether they have a spatial profiles of the gene expression to improve the population study. They call it archetype, so it's points of those cloud shape. Okay, so if you improve the correlation between genes, then you can improve this transcription factor and target prediction, because before you cannot see the relationship between the upregulation of transcription factor and their upregulation or downregulation of the target genes, but now you can recover this relationship. Well, let's before break, I want to use this example to show you the potential applications for KNN in spatial transcription. So I'm loading example data here. So here is one of my tissue slide. And then, so just rotate a little bit. And here, this slide here, and here is this 10x genomics visual, and have a probe and detect expression level onto this. Okay, so for instance, you can look at the gene expression for some of these genes. So for example, this VIM gene and express the distribution on this tissue slide looks like that. Well, one of the problem I want to study is to see, okay, so what are the genes expressed around this particular structure of this oval shape? So I want to find the genes expressed highly around this shape, but lower away from this shape. So what I'm going to do is to create a network. So I will create a network for all the spots we have sample from, and then I will compute the distance from all the spots to this shape. Follow this KNN network. I don't want to use Euclid distance because if you have a shape, a curved shape, and for example, from here, this point here to the shape, you cannot go direct distance. You have to move along this tissue and compute a more realistic distance. So what I can do here is to look at my cells and then compute. Okay, and then, so it depends on how many Ks I want to use. So this is my graph. So I can move from any point. And I have to define a region here, and that oval shape, and then to define the distance from all the data points to here, not by Euclid distance, but by the steps. It will move from one place to another. In that way, I can later, maybe I can find some genes express highly when they close to this shape, but lowly when they are away from the shape. So if you consider this particular spot in this tissue is a cancer, in situ cancer structure, and you want to look at what's the neighbor cells expression profile, what's the which gene express highly when they close to the center of the cancers. So that's the kind of applications I'm thinking about using KNN in this spatial structure relationship. Okay, let's take a 5 minutes break.
Dr. Kai, I had a question.
Yeah.
So I understand what the isomap thing represents. In a sense, it's trying to find the geodesic distance.
Yeah.
But what about the Laplace eigenmap? Is that because to me that just seems like an abstract representation of the manifold in arrays, right? Or using arrays. Right?
Yeah.
Okay. And then is the solution to that the same? It finds the shortest geodesic distance?
The solution for that construct distance matrix slightly different. The first one, you have to have this geodesic distance construct, the distance matrix, because isoform map start from a distance matrix and do FDS. Well, the second one, you play with the network. It's not directly the distance matrix. Your object you're working on is the network matrix.
Right.
They're related, absolutely related.
Okay.
You just approach the goal using slightly different methods.
Right.
Yeah.
Okay.
Okay, let's continue to mention this manifold alignment. So this concept was introduced in 2003. It's adding this manifold constraint. Manifold constraint means you have to consider your data sets. Okay, now we have two data sets now. We have to consider the follow some of the manifold structure in the high dimensional space. And they shared this common manifold, so you have to project both of the data set to the same common manifold, and also when you reduce the dimensionality and the relationship between data points had to be kept. Okay, so here is an example. So you have this structure, this data. Okay, so your data contains many several different angles picture for this teapot. We will have a same. There's nothing similar, but there's a structure similarity in the manifold level, and this is rotation of this man body object. Okay, so so this manifold concept provide this completely shared representation of the data, and allow you to reduce the dimensionality and keep their aligned of their relationship when they was in the high dimension. So this is a class of machine learning algorithms that produce projections between sets of data. Okay, so here are the two sets of data at least. Given the original data set lie on the by default. So that's our assumption. So the idea of by default alignment is we have to introduce some kind of relationship between data sets. So your input have to be one matrix A. Okay. You want to align with matrix B. You have to introduce another matrix which is a relationship between them. So there's a correlation. Okay. Corresponding matrix here. And here you have to fill that in with the C and the transposed. So in order to keep this shape. So this is a combined, this block matrix is our foundation of by default alignment about data A and B, two data sets. Okay, so this assumption is here, and we need to have a relationship, knowledge of the relationship, which is in the corresponding matrix to reflect the relationship between data points, between A and B. The projection also keep its original nonlinear relationship between data points. So here you have data A, X, B, and you have relationship between data points color coded, and they're shared by default, and then projection, and also keep original shape, not the original shape, the original relationship between data points. Okay, any comments here? Any suggestion on about the questions? I mean, comments and suggestions about your immediately thought about the applications of this. Do you think you had have some idea about applications?
Would a possible one possibly just be for maps and navigational surfaces where you would
system. So you here you have to, when you say alignment, you have two different data sets. So what's your data set one, A and B?
Like in the context of single cell data, would A and B be different samples? Or
Yeah, that's I believe that's a good idea. So if you have single cell data, RNA data from humans and another from a mouse, that's a great idea, I believe. Yes, from a mouse. Yeah, so in this case, you assume the cells, if they collected from the same organ, the same tissue, have some kind of shape which should be alignable. Yeah, good. This is an application of protein structure, and this protein structure was solved in a completely different scales. And you see red and the blue, blue is much smaller, but once they align the shape that's shared. Yeah, we can see a lot of applications. Actually, if you have two samples, you want to apply this idea and assume this complex data set you collected from two samples, share this common manifold, and then you can create a relationship to construct the corresponding matrix and help them to align and apply those algorithms to align, for example, different cell types cross samples. Okay. In this case, in single cell study, we have now so-called multi-omics approach or multi-model. So you can actually get information for the same cell, either expression and like T cell, B cell receptor, and some surface protein marker expression, and also epigenetics signals. This all become possible. So if you, for example, in this case, you have same population of cells, put them into half-half, and then one goes to single cell RNA, you get the expression matrix. Well, another you put into epigenetic study. The single cell attack-seq approach, for example, can give you the information about each individual cell's open protein structure and the promoter openness, some kind of signal. So you get the two matrices and assume they from the same population. Their underlying structure of relationship between cells will be the same, but you just use different signals to represent those cells, so they are alignable. So you can introduce some kind of a corresponding matrix and then align them. Yeah, so this is one of the applications. A quick introduction of attack seek, if just in case some of you maybe from a engineering background, you know nothing about epigenetics. So attack seek is a technique which you can use sequencing technology to measure the open chromatin structure, because the chromosome is formed in the chromatin. So the chromatin have a closed states and open states. When it's open, it's ready to either transcripts or ready to be bind or regulate other transcriptions. So this is dynamic process. Depends on the cell states. So different regions of chromatin will be open or closed, and this binding site will be accessible, and this accessible will change the state of regulation of regulatory elements and promoter activity and associated regulation at the transcription level. So it's very important if we can have a sequencing technology to know which part of the region of the chromatin is open, which part is closed. And this transposis technology allow us to have access to this small sequence. If you sequence a lot of this, at the end, you know this particular region is open. Otherwise, it's become inaccessible and you don't have a lot of reads to be read out. And now we can do this at a single cell level. So you can have a profile of each individual cell, see which part of the chromatin is open or not. That's single cell attack seek. Originally, attack seek was used on the bulk samples. Now you can have this information and you get the matrix with the cell, and essentially most commonly are the nucleus, because this chromatin is inside the nucleus. So each nucleus is a cell, and then each column here is a peak, which peak has a location in the chromatin, is which chromosome and which position. And the height of the peak is a combination of this basically yes or no answers, the binary data. Or you can have some levels of read, but combinedly you can have, see, okay, in this position, for this group of cells, you have a peak at a certain level, indicate the chromatin position at here is widely open. Otherwise, some of this location have a low peak that indicated at the population level, very few cells have this particular chromatin positions open. So you can use this to as an information to reconstruct the population cell population profiles, and also those peaks also can help you to identify the cell types. Okay, so the TC plot you get from here should, if you do the same sample for the same population of cells, use a single cell RNA-seq, you should have the same kind of population of a cell, but you construct the information, construct the TC using expression data, you should have some similar proportion of cells at least, and also distribution are shareable. So that's attack-seq data, and you have individual levels of the cells, and you can combine them to give a stronger signal, and that's the distribution of peaks along with the annotated genes. You see where they are and whether they're located in the promoter region of some genes or is in the cis-regulatory or trans-regulatory element position, which where the enhancer is located. So those information should be integrated, and then we have the algorithm that using manifold alignment to align those matrices. And this becomes more important if you have even more omics. So you have not only single cell, but now you can sequence the genome. Okay, you can have isolation profilings. Common structure is what we discussed. A TAC-seq can have transcriptome, that's single cell RNA-seq. Nowadays, we have proteomics data. We can have a lot of protein concentration have been measured at the single cell level. So to integrate those for the same cells, for the same population of cell, we can use manifold alignment approach. Okay. And also, manifold alignment is not only limited to 2 samples, 2 data sets, maybe you can have 3 or even more. So some packages claim they can do this. So you can do this using CCA or kernelized CCA. Well, you can also use kernel version of manifold alignment. So here you have red data points and the black data points, and then create this KNN respectively for each of the samples. Then you found the so-called alignable point. So this yellow point is aligned with the yellow point. So you have some anchors to know how can you rotate your structure. And because KNN getting involved, so this have some limitation of the manifold structure. And then you apply this, then you align those and rotate your structure and alignable. Make them aligned, okay? This is another package, it's called Matcher, and it's claimed that you can align gene expression as a TAC-seq signals, and by assume they have a shared manifold structure among the cells. Okay, a different algorithm is called coupled NMF, non-negative matrix factorization. In this case, it's a different solution. It's not a manifold alignment based. In this case, it consider again single cell data, and also for the same population, you have single cell TAC-seq data. So you have 2 matrices. That this non-negative factorization and apply to both matrices at the same time. Also, it doing this jointly. So this couple. So you have loss function consider both gene expression and TAC-seq at the same time to try to decompose them. Assume they have shared these fractions, okay, because they collect those from the same cell population. So some of this proportion components are shared between 2 matrices. So you have this constraint that you can somehow decompose the matrix to find the major signals. Different consideration, okay? So from okay, so this another 2 example applies manifold alignment. So this some spatial spatial data construction. This is not applied to single cell data. This will use a satellite data, called the hyperspectral image of the Earth's surface for certain region, and in different seasons, maybe you have to you want to align them somehow. And this application is manifold learning applied to gene regulatory networks, but collected from different time points. Okay, for single cell attack-seq analysis, Surrey has this side package. It's an extension of the Surrey package that allows you to do QC, to visualize, to find the peaks, to combine different peaks to for the pseudo-bug coverage, and then integrate with the single cell RNA-seq data. So some of these applications using the idea of the manifold library. So this is available for pseudo Surrey package version 3, where the attack-seq data can be processed, integrated into with single cell RNA-seq data. Okay, we finished the manifold and manifold alignment. Let me know if you have any questions. Otherwise, we are going to move to Tensor. Okay, Tensor is a higher dimension matrix. Okay, so when we say, okay, we have a rank 3 tensor, that means you have a 3D array. You have X, Y, and Z. Okay, every element in this qubit has been filled with values. So you have a 3-dimensional array or 3D matrix. And the higher dimensionality in this data structure to give you more structure, high dimension, and computation, and more challenge. We're not very good to deal with high dimensional matrix. It's okay, we have a spreadsheet to work on the 2D, but for 3D it's difficult. And the tensor is not a real limited but to 3 or 4. You can have a even higher dimension for tensor. Okay, so this 1D is a vector, 2D is matrix, 3D is 3 order tensor, and you have a higher, even higher dimensionality. This example, if you have a vector is 64 vector, 64 by 188, and this is 4 by 4 by 4. Well, in the real applications, we have a lot of the tensor data. So you have image, the image was have a different 3 layers at least. If you look at your PNG image, you have a 3 layers. It's a tensor structure. And there are pixels indicate the grayscales, and also each layer is a different color channels. 3. And for the video, and then because you have a time frame, so it's easily to have a, so one one image, two image. So you have this 3 fourth order tensor. And yeah, so the satellite images and brain imaging recommended systems. So you have users and the movies and then collect data from a different type. So you have this high dimensional data. And also those data as well. So each set is one person's writing of this. Physics, people use the tensor all the time. So this is those equations here, they all tensor-based variables. Because we are dealing with 3-dimensional and also time. So it's more difficult as you have to change your source code somehow, and then also borrow the tensor-oriented or tensor-aware packages to make your coding easier. For example, here, this is okay. So because this data, you can have X, Y, and Z, different colors, and then deal with this easily. But the other way around, if you not allow Z and that this data structure is not suitable to do this higher dimension data. So, and your coding, the way of your writing the source code is different. Vocabularies and in computer science, people call number array or 2D array. In mathematics, you have scalar, vector, matrix, tensor, and this to the tensor levels. A dimensional tensor in computer science is called a dimensional array somehow. Okay, tensor decomposition is mathematical tool for data analysis to learn from the tensor structure the components which can be used to construct original tensor data sets. And if you reduce it to matrix form, to matrix version, that's something we already deal with. We deal with matrix decomposition, that is eigen decomposition, SVD, exactly that. Then we try to learn the components. They may be orthogonal to each other, but their product is your original data set. And we can use it to do the low rank approximation, for example, to learn the features of the original data set. Same idea apply to tensor if you have a 3D matrices, 3D array, and you decompose into 1, 2, 3 components. Well, it's the same thing if you have even higher levels of dimension. Okay, so here I put SVD and the tensor decomposition side by side. At your left is SVD. Here we use just two components approximate original data matrix. So you first decompose it into U, sigma V. Sigma is just two values in the diagonal. So there are just single values in there, and then you have U matrix and the V matrix, they are rotation matrix. So same thing, if you have a 3D array, then you decompose it first into this small matrix, and then second matrix, the third matrix. In the middle, you have this small qubit with three values in this diagonal position, and all others are zeros. And this three rotation matrix, when you multiplicate along with these scaling factors in the diagonal position of the qubit, and you can reconstruct roughly the same original data with some of this noise or some levels of deviation cannot captured by those. So, in order to denoise, you can say, okay, I want to discard this and only use reconstructed this to represent the original data. That's a way to do the denoise, because you throw away which cannot be explained by this structured matrix. There's a random noise. And the reconstructed 3D array supposed to be clean. Okay, so this is the different ways to write this. Use combinations of vectors. So then you have matrix, you have vector one, B1, A and B, B1, then the multiplication of this plus that plus this to give you the approximation of the original data. You can do the same thing for high dimension data sets here. The 3D that you have vector 1A, vector 1B, vector 1C, vector 2A, vector 2B, vector 2C. Depends on how many components you want to use. You stop at level of R here. Okay. Depends on the weight of each of this. And then you can see, okay, I want to throw away those slowly express components and use major components to approximate the original data. So somehow you can also denoise the original matrix, high dimensional matrix. Same idea. Okay, how can we do the decomposition for tensors? This is called CP, versus canonical decomposition method. Historically, the CP stands for either this canonical polyadic or this, and named differently in the literature, but the name is the same thing. So when we see CP decomposition, what we mean is we decompose them into this structure very similar to SVD to minimize the difference between this high dimensional value with the product of this, depends on how many Ds you have. So the goal is to minimize the difference. Okay. Well, in this case, we don't guarantee those factors or these components are orthogonal. So this is a pretty hard problem. So how the algorithm achieve this minimization problem? Try to minimize this. It uses alternating least squares method. It works on the one direction each. So step one, to the A, and then move to B, move to C, and do that again from A, B, C. So that's the way to do it. Well, still, even you use this method, there are so many parameters you have to optimize. The search space is extremely large. There's some technique people are using, some kind of randomization procedures, try to accelerate the fitting, the optimization. Well, so that's the area of numeric linear algebra dealing with tensor decomposition. So we have to leave them to those computer scientists to solve the problem. What we can do is just use a package right now. Okay, so tensor decomposition using CP decomposition is the most commonly ways for low rank approximation for the data. So you decompose them, use components to approximate the original data matrix. I mean, high data tensor. Another way of tensor decomposition is called Turk decomposition. Structure is slightly different. It will decompose the tensor into those fibers. Okay, so you have fiber, the product of those fibers to give you the reconstructed tensor data. Well, applications. In this case, it's applied to social network. So they collect information from a school, and the school have a phone network between students and their senders, the receivers at a different time collection at the different days of per month. So this time involving network collect the multi-layers of network form a tensor structure. And then we can learn from there and to find the components from the sender and from the receivers and the frequency or weight and the time of days for each components and which day, which amounts of different weights. So you can somehow to learn those components in this highly complex three-dimensional structure of tensor. So this example is from a mouse neuron study, and they put mouse in this cross-shaped maze, and then let them to start from either east, west, north, south, north corner of this. And then at the same time, they have a record for the neuron activity of the mouse brain. And then they have different trials. Okay. So you see different trials at the start from a different position or starting point, and multiple samples, and each sample have a time frame and neural activity. So this create another high dimensionality data set. They decompose the data into 8 components. Some activity for neurons, for the time trials. Trials will separate either from a start from which position. I think they have reward to this animal. So you start from this position and you can search and find the reward. So the mouse have to learn either to turn left, turn right. So those activity and their movement have been all recorded to see whether their activity in the neurons can reflect any of this right turn, left turn, or rewarding experience. So what we can find is after decomposition, they can find those interpretable components and the proportion explanation. So you have to, if you really like, you can go here and you have a time. This video will explain to you what those curves, colors indicate. You can see some patterns. You learned from the structured data really insightful to tell you what activity, which kind of activity relate to what kind of a relationship with this activity types. Okay, at the end, I want to show you these applications we developed and using tensor decomposition and Manifold language together. Start from a single cell RNA matrix, have 2 samples. So this is sample 1 and then sample 2. You can consider, okay, this may be control, that's a disease, or this is a wild type that's knockout, whatever. You have 2 data sets collected. Here we did a cell subsampling to subsample cells and reconstruct those multiple subsamples. And we treat them independently to construct the network. Each of subsamples. The subsampling is a way you can control for some of this outer layer of cells or expression data points by average cross subsamples at the end. So for each subsample, the cells, we can create a gene regulatory network. So it depends on how many subsampling you have obtained. You have these numbers of networks. So for each sample of sample 1 and 2, you have these multiple networks. If you consider them together, this is 3D plus 2 samples, the 4D. So you have a rank of 4 tensor. 4D tensor here. If we want to do the tensor decomposition, you can decompose and learn from the data and then use the top components to reconstruct those tensors and then take average cross samples and then get the average gene regulatory network for each of the samples. So here, these 2 are denoised by decompose and reconstruct the tensor. And then we are ready for the comparison between these two networks. So here we use manifold alignment to treat each of these genes as a data point and align them. Look at the distance of the gene because we know the correspondence because this gene corresponds to this gene in the same gene in the different samples. So it's easy to construct those correspondence matrix. So after alignment, we assume they already been projected onto lower dimensional space into the shared components by default, and then look at the same gene's distance. So if they will align this same gene in the two samples should be very close to each other. Those genes which far away from each other is across the samples, the expression profile has been changed greatly. So by look at the distance for the same gene in the same sample in the projection onto the shared common manifold space, we get the distance. The far away from each other is the gene had been dysregulated or differentially regulated across these two samples. Now we can rank those genes and look at their function. And this function will reflect the difference between these two samples, biological difference maybe. So this approach is more sensitive than directly compare the high or low expression because some of the genes expression level may not change at all across two samples. The only thing changes is regulation relationship between those genes with other genes. This pipeline will be able to capture those. So I demonstrate this application just show you the applications of tensor decomposition for denoise and also for alignment for reduce dimensionality and also to help to identify those difference cross samples. Okay, that's the end of the lecture. And if you have any question, please let me know. All right, if you go good, we see, okay, there's one feedback, no problem, okay. All right, if everything is good, we see each other Thursday.